This video will be about DNA translation. So remember that DNA, in the ordering of its bases, holds the information that ultimately is used to create proteins. So changes in your DNA ultimately affect the structure of the proteins that are made. And this kind of connection between DNA and proteins occurs in kind of two steps. First, DNA is transcribed into mRNA. Just recall that here. And that's when the complementary ribonucleotides are inserted across from the template strand of DNA. So we now have a copy of mRNA. And then to get that to translate into proteins requires translation. And so let's talk about proteins. So DNA is a polymer of nucleotides. And that just means that if you hook a bunch of nucleotides together, you got DNA. And those nucleotides are made up of a sugar, a phosphate, and nitrogenous base. Well, there's a lot of parallels with proteins. Proteins are also polymers, and they're polymers of amino acids, which means if you hook a bunch of amino acids together, you get proteins, or at least a polypeptide, right? A lot of amino acids. And similar to nucleotides, there's a few things that are the same for all amino acids. So they all have an amino group here, this NH2, sometimes written NH3. They all have this alpha carbon, which is this kind of central carbon with a hydrogen attached, and R group. And they all have a carboxylic acid group, sometimes written without this hydrogen here. And like the nucleotides, there's one part of the amino acid structure that changes for different amino acids. So for nucleotides, the different nitrogenous bases determined, which nucleotide it was exactly. Oh, is this a nucleotide with cytosine or guanine or adenine, right? In amino acids, it's what is the side chain, what is the R group. So kind of everything from here up is the same for amino acids, and the only thing that changes is this one R group, this one side chain. And kind of biologically speaking, there are 20 amino acids we really deal with, which means there are 20 distinct side chains for each of those 20 amino acids. So let's talk about how these amino acids are bonded together in a very simple way. So to get amino acids to bond, to form proteins, you need to form a peptide bond, shown here. And basically what happens in a peptide bond is the OH on the carboxylic acid group of one amino acid and one of the hydrogens attached to the amino group both shift. And they both kind of shift positions in a way that allows the nitrogen to bond directly to the now freed up carbon. And so this is what you end up with. You end up with that OH and that H moving off, and this carbon and nitrogen forming a peptide bond. And that's how you connect them together. So now, if you do that a bunch of times, like here, you suddenly got a polypeptide. And polypeptides are more or less proteins, right? A whole bunch of amino acids stuck together. And the cool thing in this picture down here is that you'll see that there are the R groups. And here, it just says like R1, R2. Here, you get to see some of the diversity in the R groups. And the R groups become really important because the different R groups can have vastly different polarities. So there are going to be kind of non-polar R groups like this one, but there are also ones that are charged. And you'll find ones that are negative, there are positive ones, and you'll find ones that are polar. So they're a little more charged than non-polar, but not quite fully charged. And these have a great impact on the proteins. And you can imagine that the specific ordering of the amino acids and the number of amino acids has a great effect on the structure of the protein because these R groups can be so different. And it's the interaction of these R groups, remember, so there's slightly positive and negative parts that'll float around, and you've also got the amino groups and the carboxylic acid groups sometimes can interact. And if you hook a bunch of amino acids together, they don't have a linear structure. They'll fold up on themselves, creating a complex three-dimensional structure. And it's that complex three-dimensional structure which determines kind of what that protein can do. And so here's the big statement. If we can determine the sequence of amino acids, we can determine the functionality of the protein. And we can do that with the information in DNA.